Hello everyone, welcome to CAPSA. Today's topic is liposomes, okay? Or liposomal drug delivery system. So what are liposomes? Liposomes are sphere-shaped vesicles consisting of one or more phospholipid bilayers, and these layers are referred to as lamellae. Due to their size and hydrophilic characteristics, these are the promising system for drug delivery system. So these are sphere-shaped vesicles consisting of one or more phospholipid bilayer. These bilayers are known as lamellae. Okay. So due to their size and hydrophilic characters, promising system for drug delivery. And the property of lysosomes differ depending on their lipid composition, surface charge, size, method of preparation, etc. So the property depends on lipid composition, surface charge, size, method of preparation, etc. Okay. Now, who discovered liposomes? In 1960, scientist Wengum discovered liposome. Now, what are the types? On the basis of size and number of bilayers, liposome can be classified into two categories. First one is multilamellar vesicle or MLV. Second one is unilamellar vesicle. So, first one is multilamellar vesicles. It is mainly onion-like structure. Where several unilamellar vesicles form the other inside of the other with smaller size and making a multilamellar structures of concentric phospholipid spheres separated by layer of water. So, we will understand through a diagram. This is multilamellar vesicles. Okay, these are the phospholipid bilayers. These phospholipid bilayers does not look like lines. It look like this kind of chain. Okay, for easy purpose, I have drawn like this. Okay. Now, phospholipids looks like individual phospholipid looks like this. This is the head part and this is the tail part. Okay. The head part is hydrophilic nature. So, head part is hydrophilic. And the tail part is hydrophobic. Now, what are unilamellar vesicles? Unilamellar vesicles has a single phospholipid bilayer sphere enclosing the aqueous solution. So it has only one phospholipid bilayer. Now we will understand it through a diagram. It looks like this. One type of layer. Okay. This is unilamellar vesicles. And these unilamellar vesicles again subdivided into two categories. First one is small unilamellar vesicles or SUV. This is MLV, this is ULV, and large unilamellar vesicles or LUV. They actually differs in the size. Simply in the size, this is less than 100 nanometer in diameter, and this is more than 100 nanometer in diameter. Okay. Now, what are the components? So, components. Mainly two types of components are used. First one is phospholipids. These are phosphoglycerides, sphingolipids, and phosphatidylcholine. This type of phosphoglyceride, mainly phospholipid, is mainly used. The second component is cholesterol. Now, what are the method of preparations? So, method of preparations. Two type of methods are used. First one is passive loading technique and active loading technique. Okay. So first one is passive loading technique. In passive loading technique, it is subdivided into three categories. First one is mechanical dispersion method. In mechanical dispersion method, varieties type of methods are used, like lipid film hydration, it can be handshaking method, non-handshaking method, or kind of freeze drying. And it can be microemulsification, sonication, French pressure cell, membrane extrusion, dried constituted vesicles, freeze thawed liposomes. These are the methods of mechanical dispersion type. Okay. Now we will look the second solvent dispersion method, which is another type of passive loading technique. Okay. In this solvent dispersion method, what is used? First method is ethanol injection. Second one is ether injection. Third type is double emulsion vesicles. Fourth is reverse phase evaporation vesicles. Fifth is stable plurilamellar vesicles. So these methods are solvent dispersion method of passive loading technique. Another last passive loading technique is detergent removal technique. For this purpose, which type of methods are used? Actually here, detergent removal occurs from mixed missiles by the process like dialysis, dilution, column chromatography, reconstituted Sendai virus, enveloped, etc. Okay, so these are the methods, and last one is the main method of preparation, active loading technique. It is not categorized in subcategory. Okay. Now, what are the advantages? Actually, this type of drug delivery system increases the efficacy and therapeutic index of drug. Also, it increases the stability via encapsulation. Obviously, it is non-toxic, biocompatible, and flexibility to couple with site-specific ligands to achieve active targeting. So these are the main advantages. Now what are the disadvantages? Disadvantages are low solubility, short half-life, sometimes phospholipids undergo oxidation, and hydrolysis. This type of reactions, okay? And leakage or fusion of encapsulated drug. Another disadvantage is product cost is very high. Now, what are the applications? Applications are, it actually used as vaccine carrier, as a artificial blood surrogates, as radiopharmaceutical and radiodiagnostic carrier. In bioengineering, cosmetics industry, and agro-food industry. So these are the use of liposomal drug delivery system. Okay. Now, this is the end of my lecture. If you like my video, do like, comment and share. Thank you for your attention.